Dennis talked to you about isolating antibodies from natural infection that are broadly neutralizing, and Ian showed you structures. What I'm going to talk about is trying to translate those pieces of information into immunogens that might re-elicit such antibodies. And what we spend most of our time on in my lab is, which is actually my lab, not David Baker's lab, is trying to design. That's okay. He does a lot of things, so it's hard to stay outside. And he's a great guy. Is try to design immunogens that are focused on particular epitopes. And in the next two sort of movies, I just want to give you an example from HIV about, just to bring it home about why we think that's necessary for HIV, and probably for many other very challenging viruses, you'll also find it necessary. So this is cryo-electron tomography density of HIV trimeric spike taken from Sriram Subramanian's paper in Nature 2008. And on it, we've docked in Peter Kwong's core GP120 crystal structure, 3 copies of it. So this is a trimeric spike. And wiggling around are the glycans that are only on core GP120, but so there's 16 glycans on each core, but there actually are 4 more glycans on each copy of GP41 down here, so 12 more down here. And then 6 to 7 more glycans on the variable loops at the top of each monomer. So you can see already with these glycans that a lot of the spike is covered, but there are more that I'm not showing you. In yellow is the CD4 binding site, which is this conserved target for antibodies. And we know there are antibodies that target that site, but you can see from the structure that it's in a recessed cavity and it's surrounded by glycans. The variable loops that we know are immunodominant are protruding through the glycan shield at the top, attracting immune responses and escaping it whenever HIV infects an individual. So you can imagine, even if you could take a perfect trimeric mimic and design a vaccine that way, like Dennis was talking about, have been done so commonly for many other viruses, it probably wouldn't work. You probably wouldn't get antibodies directed against the CD4 binding site. No one has made a perfect trimeric mimic yet for HIV, so we don't know for sure, but probably we would get a lot of antibodies to the viral, to the variable loops. And so this structure says to you, well, you know, what can you do? What can you think of in terms of trying to re-elicit responses that we know humans do produce? And just to keep bringing it home, you know, we do know that antibodies do access these epitopes. This is cryo-electron tomography density of 3 copies of B12 antibody in purple and blue bound to its conserved epitope in the CD4 binding site. And the glycans will turn on. You can see, you know, it just squeezes right into its epitope. The trimer actually is a little bit more open when B12 is bound, and whether that reflects, you know, some kind of induced fit or if the trimer is breathing on the surface, we don't know. But the problem is, if you immunize with a GP120 subunit, or you make trimeric mimics in all the last 20 years, no one has really elicited B12-like antibodies. And so what are we going to do? And this all falls in the realm of what Dennis was talking about. I usually show this figure from his paper. It's not clear what we call it now, reverse vaccine design or reverse vaccinology. Basically, the idea is you isolate antibodies from natural infection that are protective, and you characterize their interaction with their antigens, sort of just in the way that Ian was showing you by crystallography for flu, and then you use all this analysis from crystallography and other measurements to try to do immunogen design, and that's sort of where we come in to try to manipulate the structure of the antigens and try to devise antigens that will re-elicit antibodies against specific conserved epitopes. You know, we're spending most of our time on HIV. We've been talking to Crucell for quite some time of trying to translate this information to make, you know, to help make a universal vaccine for flu, and we're excited about that. I also have some work specifically on RSV in my lab that's really exciting, that is all unpublished and unsubmitted, so I'm not going to talk about it here, but you know, stay tuned to this channel. So the first, I'm just going to restrict my comments to 2 sort of strategies that we've published already, but you know, there's quite some interesting results, and draw sort of parallels to what could be done in other cases. So the first place that we started work were these 2 epitopes from HIV that occur right at the base of the spike. I showed you this twirling spike, and these occur right at the base near the viral membrane, similar to the way that the 8020 epitope on hemagglutinin occurs near the membrane. The 2F5 epitope is this yellow, sort of poorly defined squiggly structure, and the 4E10 is a sort of a nicer helix. And these have been studied for a very long time. I mean, these are some of the earliest antibodies isolated against HIV. They're not as potent as the new ones that Dennis talked about, but their 4E10 is perhaps the most broad if you're considering low levels of antibodies still. And, but the problem is, no matter what people have done with these epitopes, you simply can't re-elicit these antibodies. You don't elicit broadly neutralizing antibodies at all. And 4E10 in particular is very hydrophobic, and even if you take this peptide and put it into variable loops of GP120 and immunize, you don't even get response against the peptide. And so we started in collaboration with several other people trying to do a strategy of scaffolding where we transplant the structure of an epitope onto a protein scaffold that's already folded and so already looks the way the bound conformation of the epitope looks. The idea being that now if you can use this as your immunogen, maybe if this is fixed rigidly or relatively rigidly, you might have a better chance of re-eliciting structure-specific antibodies. And if you can do this by computational design or in vitro evolution or a combination of the two, you may be able to devise a whole bunch of different scaffolds for the same epitope, and that can open up heterologous prime boosts or cocktail experiments that may also allow you to focus. You can imagine manipulating the structure of these a lot to try to learn how to focus the response. And so we started off with both of these, with projects on both of these, and I just wanted to summarize where we got so far. So we published some papers last fall, and for the 4E10, which I mentioned was a very poorly immunogenic epitope, and Dennis and Ian had one of the most intelligent previous efforts where they, as I mentioned, they put the epitope into the variable loops of GP120 and they rotated the epitope around to optimize the exposure, and they didn't get any immune response. Here we designed a whole panel of different scaffolds, and Roland Strong's lab solved crystal structures of them unliganded, showing very perfect mimicry to the bound conformation of the epitope, and also liganded, illustrating that the antibody is binding the way we thought it should in our model. And when we immunized rabbits with these scaffolds, we published results for one of them so far, and this graph is showing a comparison of the epitope specificity of the polyclonal response induced in rabbits compared to the epitope specificity of the 4E10 monoclonal itself. So this is Luminex data looking at the binding of 4E10 in black to alanine mutants of the peptide, and you can see the wild-type peptide, it binds with a relative avidity of 1. If you mutate this W672 to alanine, the binding goes to basically 0, so 4E10 requires that W. If you mutate this phenylalanine to alanine, again the binding goes down. But if you go down to positions that are on the underside of the peptide that 4E10 doesn't engage in the crystal structure, you see if you mutate this asparagine to an alanine, 4E10 binds just as well as it does to the wild type. It doesn't care. And this all makes sense from Ian's crystal structure that he had previously published. But the polyclonal rabbit sera tracks very similarly. The blue and the red bars are from immunizations of rabbits with the same scaffold using two different adjuvants. And you can see the blue and red bars track roughly pretty well with the monoclonal itself. So we were quite excited that not only did we elicit antibodies against the epitope, which really hadn't been done to any strong degree previously, they looked like they had the right structural specificity. And then on the 2F5 project, we had essentially similar results. Peter Kwong's group solved crystal structures of a scaffold bound and unbound, and it looked the way it should look. If you immunize rabbits, again, now this is ELISA data against alanine scanned peptides, and in black is the response of 2F5, and so if you mutate this D, K, or W to alanine, the binding goes to 0. It's really required for binding this antibody, and this was our best polyclonal response from rabbits, which tracks really closely. And just to compare, a very traditional approach would be to take the peptide and conjugate it to KLH and immunize. And so Peter and Adi did that, and Rich Wyatt did that. And the red traces here are the polyclonal responses from rabbits that were immunized with KLH peptide. And you can see, again, in black is the 2F5 response. The polyclonal responses from peptide immunization are completely different from the desired pattern and much less similar than the responses elicited by scaffolds. And Peter and Adi went one step further, and they they made mouse monoclonals from mice immunized with a heterologous prime boost of the scaffolds that I designed, and they solved the crystal structure of several of those mouse monoclonals bound to the 2F5 peptide. And in this paper, there's a figure that shows 2F5 in the surface here, bound to the 2F5 peptide, and this is 11F10 is one of these mouse monoclonals elicited by the scaffolds in the surface, bound to the 2F5 peptide. And you can see the electrostatics of the antibodies are similar, and the bound conformation of the epitope is quite similar. And just to show in this figure, if you just superimpose them, this is the mouse monoclonal in sort of red and blue. In this long white or gray structure is the bound conformation of 2F5 peptide, and in yellow is the bound conformation of the peptide as bound to the mouse monoclonal. You can see here very clearly how similar the bound conformations of the epitopes are. And so this is sort of a direct crystallographic demonstration that we re-elicited a specific structural response. So we were quite excited, and so the initial conclusions about these artificial immunogens are that they do have utility as immunogens. They can precisely mimic viral epitope structures, at least we've shown for continuous epitopes like 2F5 and 4E10, and they can elicit antibodies against those epitope structures even for poorly immunogenic epitopes like 4E10. The caveat to these studies, and the reason that they weren't in Science or Nature, is because when you're doing vaccine design, if you don't elicit neutralizing antibodies, everybody said, well, you didn't do any better than anything before, and so, sorry. And in these cases, the polyclonal responses weren't neutralizing, and obviously that was a disappointment, but very early in our work and continuing during our work, other people, including my collaborators, spent a lot of time characterizing the mechanism of neutralization for both of these antibodies, and they showed that it requires more than simply interacting with the peptide epitope. And I'm just going to take you quickly back to explain this. So in both of these cases, 2F5 has a long HCDR3 loop that's here, and 4E10 has a long HCDR3 loop that's hard to make out, but it's tucked back to its structure. And in each case, there's a multiple hydrophobic residues, but in both cases, there's a hydrophobic residue on the tip of the HCDR3. And if you mutate those to alanine, the mutant antibodies still bind to the peptide with very, very high affinity, but their neutralizing potency drops by huge factors, and it's strain dependent. And so that shows you that when the antibody is interacting with the virus, the HCDR3 is doing something that is not evident from these crystal structures of the peptides. And our scaffolds, it may be a little hard to tell, but the way we designed them, we didn't know that the HCDR3 was important. And so our scaffolds really don't touch the HCDR3 loops of either antibody. And so we didn't give those rabbits any reason to make a specific HCDR3 like 4E10 or 2F5. And that's our, Scott said you should always have your hypothesis and you should chase after it. So we are chasing after those things now to try to go one step further and have scaffolds that will elicit neutralizing antibodies through these epitopes. So that's one key open question, is can we use these artificial immunogens to elicit neutralizing antibodies? And we're testing that in these cases and in other cases that I won't cover here. But another general question about these sort of artificial immunogens is from the perspective of vaccine design, most epitopes are complex. They're not linear epitopes like 2F5 and 4E10. And it would be much more useful to be able to study the rules of immunogenicity if you could design scaffolds for discontinuous epitopes. And so we set out to try to transplant. I showed you in my second movie the cryo-electron tomography of 3 copies of B12 bound to the CD4 binding site, and I explained that really no one has elicited that antibody except for pulling it from libraries of natural infection. But there is a crystal structure of B12 bound to GP120, and we set out to try to transplant key parts of the B12 epitope onto non-HIV scaffolds to then allow us to see if we could use those scaffolds to do a better job of re-eliciting B12. And I can't go into a lot of detail about this because we've submitted a paper, but we did manage to succeed in transplanting this epitope, and we have a crystal structure of it bound, and it reproduces the interaction quite precisely. And we're now trying to see what are all the different ways we can use these kinds of scaffolds to to see if we can re-elicit B12-like antibodies, which would be a huge advance if it would work. Then I just want to touch on one other sort of technology that has been useful already in publications, and I think potentially could be helpful in terms of immunofocusing. And this is the idea of antigen resurfacing. It's a pretty simple idea. It's been around for a while. We started, in a conversation with Peter Kwong and George Shaw, I suggested it for GP120. And the usefulness there was the idea that if you had GP120, and here's yellow for the CD4 binding site, and here are the blue glycans, if you could change all the amino acids on the surface outside the CD4 binding site, you could imagine making a variety of these molecules and then using a cocktail or heterologous prime boost sequential immunization to focus the immune response on the one patch that doesn't change. And we set out to do that in collaboration with the VRC. Them doing most of the work, me doing the design, but them doing all the wet lab work and characterization. And the work for immunization is still underway. But an early side benefit was Gary Nabel and John Mascola had a very clever idea of using, and Rich Wyatt had a clever idea of using this resurfaced GP120 as a probe for pulling out new antibodies in the way Dennis told you. And this was published last year. And so that molecule that was resurfaced that I designed was used as a probe for B cells, and they pulled out this antibody VRC01 that binds to the CD4 binding site just like B12 does, only it's far more potent and far more broad than B12. So it was kind of a nifty side benefit that structure-based design assisted in pulling out this new antibody, and now this new antibody can be used as a template for further structure-based design of hopefully of a vaccine. And along those same lines, we've been pursuing resurfacing and sort of remodeling of antigens in a more generic way. And we published this paper as a sort of as a test case where we had one of our scaffolds for 4E10. Here's 4E10, here's our scaffold. The epitope is in red. I'm almost done. And in our very early tests in guinea pigs with the wrong adjuvant, actually, this scaffold didn't elicit 4E10-like antibodies. It turns out with some more reasonable adjuvants it does, but initially we thought this was really a problem, and we speculated that we could get rid of this domain and maybe it would help the immune response. So we made a kind of a fancy computational protocol to get rid of this and rebuild this region with the helix. It includes rebuilding the core of the protein to eliminate that domain and maintain a stable structure. And then we went on to completely resurface the exterior of that protein. And I can't really talk about immunization of those things yet because we haven't published that work yet. But I can tell you that we solved the crystal structure of our TRIM, and it agreed basically exactly with the design. The trimmed proteins had much better thermal stability compared to the parent. The melting temperature went from 44 to 79 degrees, which probably is a useful thing in a vaccine generally. So that's an example of where structural-based manipulation, even if it's not directly improving immunogenicity per se, if you can avoid the cold chain, or this may indirectly improve immunogenicity by making it more stable. And it maintained very high affinity for E10. But one of the coolest thing was we had guinea pig sera that had been elicited by the parent by this protein. And if you put the trim on an ELISA plate and you ask, does the guinea pig sera bind to the trim, it binds very well to the parent, but it binds much less well to the trim. So it does look like in this case, by eliminating this domain, we did eliminate an immunodominant domain. And so we're working on immunizing with these molecules to see if they can increase the response to the 4E10 epitope. So just in conclusion, we're trying to figure out what are all the different ways we can think of how to translate structure of an antigen into immunogenicity. And there's a lot of things to talk about, and I won't go through them all. But essentially, we try to stabilize the antibody-bound epitope conformation when that's known. We expose that conformation to the immune system, preferably without immunodominant variable epitopes present on viral envelope, or you can imagine on any sort of variable pathogen. And we try to dampen off-target epitopes by various methods. And this is just a slide to illustrate how we go about collaborating with a whole bunch of different other types of labs in the pursuit of vaccine designs. And so, you know, Dennis talked about antibody discovery, and Crucell is an expert in that area. Then there's crystallographic analysis, and Ian gave you a talk about that, and then there's other people working on that. We're working on computational design and in vitro evolution of antigens, and Ian mentioned David's lab working on even computational design of antibody mimics, if those could be used. And then there are these cycles of development where, you know, express and purify and characterize these designed immunogens crystallographically. This is sort of a faster cycle. This is a little bit of a slower cycle. And then there's immunological analysis, which is the slowest cycle. And so I would say the limiting factor really is this arrow. Limiting factor is getting this data and feeding back to revise the design. And there is some effort, you know, in terms of in vitro immune systems and predicting immune responses. But for a company like Crucell, I would say this would be one area to seriously think about investing in and doing some really novel work in terms of, can you predict the immune response to a given protein? Can you rapidly evaluate something without waiting for the animals all the time? Because unless you have this in the lab, you don't go to the clinic, and the clinic is where we all want to go. So that's, I'd like to thank funding from the Gates Foundation and the IAVI Neutralizing Antibody Consortium, and a fellowship for my student Bruno from the Portuguese government. And, you know, a large number of people are involved in this work, a whole bunch of people in my lab. Bruno and Mihai on the B12 scaffold, Chris Carrico solved the crystal structure, Andrew Ban on the 4E10 scaffolds. On the 2F5 scaffolds, it was really Adi Ofik and Javier Guanaga working with myself, and Rich Wyatt and Peter Kwong. I'd like to thank David. He's been a great mentor all along. On the 4E10 scaffolds, it was all done with Leo and Roland and people in their lab. Thank you.